My name is Carrie Marguiles, and I'm a sixth-year graduate student in the Department of Biological Engineering, and I am in Leona Samson's lab. The Samson Lab studies the effects of DNA damage at all levels, from molecular level up to organismal studies. We studied the role of various DNA damage repair proteins and how modulation of their activities can lead to differences in a cell's response to DNA damage, such as the DNA damage caused by chemotherapeutics used to treat cancer. One of the DNA repair proteins that we study a lot in our lab is called AUG, or alkyl adenine glycosylase. This repair protein searches the genome to find damaged DNA bases. It then finds them. After it finds them, it cuts them out of the DNA strand and recruits other proteins to finish the repair to get a fully undamaged DNA strand. And in our lab, we study how changes in the level and activity of AUG will alter your response to DNA damaging agents. So this image shows pluripotent stem cells which have been induced to differentiate into neural tissue. So we start with mouse stem cells, which are pluripotent, which means they can differentiate to become any type of mouse cell they want to. And we allow them to aggregate and create clusters of cells called embryoid bodies. And these embryoid bodies grow similarly to how an embryo would grow as a mouse develops in utero. We induce them and push them to become neurons by treating them with various cytokines and chemokines that will help them go down the neural lineage and become neurons. So in the image, the cluster, the fuzzy cluster on the side is the remnants from the embryoid body, where you see a lot of blue staining, where each blue dot is a specific nuclei of a different cell. And emanating from the embryoid body are different cell types. So in the green, you'll see axons of mature neurons, and in the red, you'll see staining of mature astrocytes, which have differentiated from the embryoid body. This is interesting to us because, as I mentioned, we see differences in sensitivity when you lose the AUG protein in stem cells compared to neurons. And so we're developing and optimizing the system as a model system where we can study in the stem cells, loss of AUG leads to sensitivity to alkylating agents, and then over a 3-week differentiation period in culture, you should get neurons that are now resistant to alkylating agents when you lose AUG. And so we want to do this to understand what makes these two different cell types different, other than the obvious differences, one is a neuron and one is a stem cell, but why is it that initiation of the repair pathway leads to repair and healthy cells in the stem cells, but initiation of the repair pathway in neurons actually leads to sensitivity in the face of DNA damage? But really, when you see images like this that work, it's highly motivating because, one, they're just beautiful to look at, independent of what the science is. But two, they're really going to be helpful, and they're going to give you the cell types you need to understand the biology behind how changing the level of the DNA repair protein is really changing the response of the cells to the DNA damaging agent.